अस्सलाम वालेकुम। तो हम डिस्कस कर रहे थे द hallmarks of the cancer, in which the next one is evasion of the apoptosis. Normally, whenever there is a damage to a DNA occur or the cell become mutant, the cell undergoes the apoptosis or the cell death. But in new plastic cell, there is no apoptosis occur, or we say that there is evasion of the apoptosis occur because the new plastic cell that involve mutation in the gene that initiate the process of the apoptosis. Normally, there is a balance between the proapoptotic and the antiapoptotic gene, but during the cell NG, when the stress on the cell increases, there is activation of the proapoptotic gene and the inhibition of the antiapoptotic gene, and hence they will lead to the process of the apoptosis. But a new plastic cell, there is gain of function mutation in the anti-apoptotic gene and the loss of function mutation in the pro-apoptotic gene. So when the anti-apoptotic gene are activated and the cancers go, there will be no apoptosis occur and the cell become immortal and they will divide rapidly and will leads to the new plastic changes. अब बात करते हैं about the genes that are involved in the apoptosis. They include the pro-apoptotic gene like a BAX and the BAT family, and these pro-apoptotic gene initiate the process of the apoptosis. And the next one is anti-apoptotic gene that include BCL2, BCLXL, and the MCL1 gene. And these genes they inhibit the process of the apoptosis. And the third family is BH3 family that include BID gene, Puma gene, and the NOXA gene. And these BH3 family they maintain the balance between the pro-apoptotic and the anti-apoptotic gene. And similarly, this BH3 family has a slight pro-apoptotic activity also. अब आ जाते हैं about the pathways involved in the apoptosis. So it include the extrinsic pathway and the intrinsic pathway. The extrinsic pathway is also known as the receptor ligand pathway or the PAS ligand pathway, and this pathway is activated by the tumor necrosis factor alpha. And the second one is the intrinsic pathway. It is also known as the mitochondrial pathway, and it is the commonest pathway involved in the apoptosis. And the intrinsic pathway is the most common dysregulated pathway in the cancer. Whenever there is decrease in the growth factor stimuli occur, or there is increase in the concentration of the reactive oxygen species, chemicals or radiation, or there is increase in the endoplasmic stress occur. All of these changes causes damage to the DNA. And whenever there is damage to a DNA occur, this DNA damage is sensed by the p53 gene. And when the p53 genes are activated, they either repair the DNA, or when there is no repair occur, there is activation of the polyapoptotic genes like BAX and the BET family. And when these polyapoptotic genes are activated, they increases the permeability of the mitochondria to the cytochrome C and the secondary mitochondrial activator caspases. So whenever there is increase in the mitochondrial permeability occur, there is release of the cytochrome C occur, and the cytochrome C increases the activation of the apoptotic activator factor 1, and this apoptotic activator factor 1 causes activation of the caspase 9, and the caspase 9 then further activate caspase 3, 6, and 7, and these caspases leads to the activation of proteases and endonucleases, which causes the breakdown of the cellular components. Similarly, there are other proteins like inhibitors of apoptotic proteins. These inhibitors of apoptotic proteins, they inhibit the caspase 9 and hence they prevent the process of the apoptosis. But due to increase in mitochondrial permeability, there is also release of the secondary mitochondrial activator caspases, and this causes the inhibition of the activity of inhibitors of apoptotic protein. And when the secondary mitochondrial activator caspase inhibit this protein, there will be no inhibition of the caspase 9 and the process of apoptosis will occur. अब बात करते हैं about the mechanism of the apoptotic evasion, or how the new plastic cell they evade the process of the apoptosis. So it involves three mechanisms. First is loss of function of p53 gene. Second is activation of inhibitor apoptotic protein, and the third one is overexpression of the anti-apoptotic gene family. So the first one is loss of function of p53 gene or inactivation of p53 gene. The p53 gene is inactivated due to a loss of function mutation in p53 gene. There is overexpression of the MDM2 protein. The MDM2 protein they bind with p53 gene and causes the degradation of the p53 gene. And when the p53 gene is inactivated, there will be no induction of the apoptosis occur. The second one is activation of the inhibitor of apoptotic protein. These inhibitor of apoptotic protein they bind with the caspase 9 and they inhibit the caspase 9. And when the caspase 9 is inhibited, there will be no apoptosis occur. And when the apoptosis is inhibited, the cell become immortal and will lead to a new plastic changes. The third one is overexpression of the anti-apoptotic gene family. The anti-apoptotic gene family, they include the BCL2 and the BCLXL family, and these anti-apoptotic gene, they inhibit the process of the apoptosis, and the overexpression of the BCL2 family, it is commonly seen in the follicular lymphoma. Normally, the BCL2 gene, which is an anti-apoptotic gene, it is present on the chromosome number 18. But in follicular lymphoma, there is translocation of the BCL2 gene from chromosome number 18 to the chromosome number 14, where the BCL2 gene combined with the Ig gene. Normally, the Ig gene is active all the time and it will express. So, whenever there is a translocation from chromosome number 18 to 14 occur, there is formation of the BCL2 Ig hybrid occur. And this BCL2 Ig hybrid overexpressed and it is active all the time, and hence there will be more production of the anti-apoptotic gene or the BCL2 genes, and this BCL2 gene overexpression will lead to the inhibition of the apoptosis and the cell become immortal, means that there will be no death of the new plastic cell occur and will lead toward the malignant transformation or the new plastic changes. Similarly, in new plastic cell, the BCL2 is overexpressed are these overactivity of BCL2 occur because of the loss of function mutation in the microRNAs. Normally, the microRNAs, they are produced and they inhibit the anti-apoptotic genes like BCL2 gene. But whenever there is mutation, loss of function mutation in the microRNAs, there will be no inhibition of the BCL2 family, and the BCL2 will overexpress. And when they overexpress, there will be no apoptosis occur and will lead to the new plastic changes. So the next hallmark is the cellular metabolism. As the new plastic cell, they grows rapidly, so they do not rely on the normal cellular metabolism. So the new plastic cell, they shows alteration in the cellular metabolism. In normal cell, there are two pathways for the ATP synthesis. One is the cytoplasmic glycolytic pathway or glycolysis, that produces 2 ATP, and the second one is mitochondrial oxidative phosphorylation, which produces 36 ATP. But in cancerous cell, the cell growth is more rapid, so they require more proteins, lipids, and nucleotide for the cellular growth. So in cancerous cell, the glycolytic pathway predominate, and the oxidative phosphorylation is the less efficient pathway in the cancerous cell. The glycolytic pathway it predominate in the new plastic cell because the glycolytic pathway or the glycolysis, they provide intermediate for the synthesis of the lipids, proteins, and the nucleic acid, which are required for the rapid cell division and the cell growth. The next one is the Warburg effect. According to this effect, the cancer cell, they shows increase in the glucose uptake due to the overexpression of the GLUT receptor, which causes increase in the absorption of the glucose into the cancer cell. Similarly, in cancer cell, there is increase in the glycolysis occur due to overexpression of the glycolytic enzyme. And there is also increase in the lactic acid production occur. And new plastic cell, there is reduced oxidative phosphorylation occur. And in new plastic cell, there is an increase in the ATP synthesis occur to the substrate level phosphorylation, in which the ATP is synthesized directly from the substrate. So the increase in the glucose uptake by the new plastic cell is known as the hunger effect. And the clinical significance of hunger effect is that it is used for the visualization of the tumor cell to the PET scan. So we give the 18-fluorodeoxyglucose, which is a non-metabolizing derivative of glucose, to the patient having the cancer, and this 18-fluorodeoxyglucose is taken up by the tumor cell and the cancer cell, which is then observed to the PET scan and give positive result. So the increase in glucose uptake is also used for the diagnosis of the tumor cells or the cancer cells. So the tumor cell and the rapidly growing cell, they rely mostly on the glycolytic pathway. And a rapidly growing embryonic cell, when the desired growth is achieved, the aerobic glycolysis stops. But in cancer cell, the aerobic glycolysis continues due to the loss of function mutation in the tumor suppressor gene and the gain of function mutation in the proto-onco gene. The next one is the cancer cell. So the cancer cell, they require limitless replicative potential to three pathways. The first one is the via the phosphoinositol 3 kinase pathway or the AQT pathway. This pathway, they upregulate the activity of glucose receptor and the glycolytic enzyme. This pathway causes increase in the synthesis of the GLUT receptor, which increases glucose absorption, and similarly they also activate the glycolytic enzyme, hence they increase the process of the glycolysis. And due to increase in the glycolysis, there are more glycolytic intermediates form, which are required for the protein synthesis, lipid synthesis, and the nucleotide synthesis. And all of these three are involved in the rapid cancerous cell growth. The second one is the cancer cell also to the tyrosine kinase pathway. Then this pathway they inhibit an enzyme known as the pyruvate kinase enzyme, and this enzyme is involved in the last step of the glycolysis. The pyruvate kinase that causes the conversion of phosphoenol pyruvic acid into the pyruvic acid. But when this kinase enzyme, pyruvate kinase enzyme is inhibited, there is no formation of the pyruvic acid occur, and there is accumulation of the more glycolytic intermediate. And these glycolytic intermediates are used for the DNA synthesis, RNA synthesis, lipid, and the protein synthesis, which are required for the rapid cell division. Similarly, a cancer cell they also act via the MYC pathway, and the MYC they increases the uptake of glutamine into the cell. Similarly, this pathway also increases the glutamine metabolization, and hence they promote the synthesis of the various cellular components which are required for the new plastic growth. The next one is the oncometabolism. So the oncometabolism occurs in the cancerous cell. According to a tumor genomic sequencing studies, we state that the new plastic cell they consist of mutation and certain enzymes that participate in the kill cycle. The most common enzyme mutated is the isosaturate dehydrogenase. Normally, this enzyme causes the conversion of isosaturate into the alpha ketoglutarate in the Krebs cycle. But when this enzyme is mutated, this causes the mutant form of isosaturate dehydrogenase causes a conversion of isosaturate to the 2-hydroxyglutarate, and this 2-hydroxyglutarate this act as an oncometabolite, and this 2-hydroxyglutarate causes excessive DNA methylation and the histone modification, which leads to the alteration in the cancer gene expression and causes new plastic changes. And the isosaturate dehydrogenase mutation is commonly seen in the cholangiocarcinomas, gliomas, and acute myeloid leukemias. The next one is the relationship between the cancer cell and the autophagy. In case of severe nutrient deficiency, first there is arrest of the cell growth occur, and then the cells start eating their own organelles for energy production and cellular survival. And this process of self-eating of the organelle is known as autophagy. And autophagy is a kind of a cellular adaptation. If there is no autophagy of the cellular organelles occur, then the cell will not survive and they undergoes a cellular death. But a tumor cell, they do not trigger the process of the autophagy because the genes that control the autophagy are the tumor suppressor gene, and these genes are mutated in the tumor cell. So the tumor cell, they do not undergo the autophagy. And autophagy has two effects on the tumor cell. If the tumor cell they undergoes autophagy, then they enters into the dormant phase. And dormant phase, the tumor cell are in inactive form, and these tumor cells they do not respond to the therapy. So which leads to the therapeutic failure. So the autophagy may causes therapeutic failures in the cancer cell. Similarly, if the autophagy occurs, they causes the arrest of the cellular growth, and hence due to autophagy, the tumor cell will not grow. And the next hallmark is the limitless replicative potential. As the new plastic growth consists of the cells that divide continuously and they have limitless replicative potential, so we say that the cancer cells are immortal and they divide continuously and they do not die. About the mechanism involved in the limitless replicative potential. The first one is the evasion of the senescence. Senescence means arrest of the cell cycle. As a new plastic cell, there is no arrest of the cell cycle occur, so we say that there is evasion of the senescence occur in the cancer cell. As a normal cell, they divide up to 60 to 70 times. After that, there is shortening of the telomere occur, and the cell they undergo the cell cycle arrest and they enter into the senescence stage. And the cell cycle arrest depends upon the p53 gene. If there is activation of the p53 gene, they arrest the cell cycle at the G1S phase, which is the first checkpoint. If there is loss of function mutation in the p53 gene, then there will be no arrest of the cell cycle occur and the cell will continue to divide and lead to the new plastic changes. The second mechanism involved in limitless replicative potential is evasion of the mitotic crisis. So in new plastic cell, there is evasion of the mitotic crisis occur. Each chromosome they consist of the telomere at the end, and telomere is a short sequence of the DNA nucleotide, and this telomere is involved in the cell division. So we say that the telomere they prevent the process of the aging. With each cell division, the length of the telomere get shortened, and after 60 to 70 division, the telomere they attain the shortest length, and after that there will be no cell division occur, and the cell they enters into the stage of the mitotic crisis, which leads to the cell death. But in new plastic cell, there is activation of an enzyme known as telomerase, and this enzyme they maintain the size of the telomere, and hence they prevent the mitotic crisis. Similarly, if the size of the telomere is maintained, they causes the rapid cell division and will lead to the cellular proliferation. Due to rapid cell division, when the telomeric DNA they evolves, there is exposure of the chromosomal ends which are sensed as a double-stranded DNA breaks. Let's suppose this is a chromosome and the cell undergoes rapid cell division, so the telomere they gets eroded and the chromosomal end are exposed. There depends upon the p53 gene. If p53 gene is active, the cancer cell they cause a cell cycle arrest and they induces the process of apoptosis. But if the p53 gene is inactive, then there will be no cell cycle arrest occur and the chromosome which have double-stranded DNA breaks at the end. They causes non-homologous end joining of the two chromosomes. Let's suppose there is two chromosomes and these telomeres are eroded. There is and the two ends they are exposed as a double DNA strands. So there is joining of the two chromosomes occur and they form a dicentric chromosome. And the dicentric chromosome form they attain multiple mutation during the cell cycle and that depends upon telomerase activity. If the telomerase enzyme which is inactive in the normal somatic cell, then the dicentric chromosome they will undergo massive apoptosis and will lead to a cell death. But if there is reactivation of the telomerase enzyme as in the neoplastic cell, then it will causes the rapid cell division and the cell will divide continuously and will lead to the malignant transformation of the cell. The telomerase is an enzyme that is active in the rapidly dividing embryonic cell and the neoplastic cell, but telomerase enzyme activity is absent in the normal somatic cell. The third mechanism involved in the limitless replicative potential is the capacity for the cell renewal. So the new plastic growth they consist of the cancerous stem cell, and these cancerous stem cell have the capacity of cell renewal and the differentiation. And these cancerous stem cell they arises from the malignant transformation of the tissue stem cell, and they also arises from the mutation in the somatic cell. And the cancer stem cell have telomerase activity which causes rapid cell division. Similarly, they are also resistant to the mitotic crisis and they have the capacity of the cell renewal to maintain its own germline. And all of these properties they make the cell immortal. And these changes they causes malignant transformation of the normal cell to the cancerous cell.